Okay.
So we're
Okay.
recording now.
Okay.
So you can go ahead and start your first take, and then just trim that off
Okay. Yeah.
afterwards.
Yeah. Yeah. But
Definitely. If you hit stop, it'll say, "Are you sure you want to stop?" Just hit OK,
right. Okay.
and then
Yeah.
that'll create each take of
Yeah.
what you're doing.
Yeah. Yeah. Yeah.
That's
Okay.
the way to go.
Yeah. I read about. I guess that happens with quadcopters. If they run out of battery and crash, you get a corrupted MP4. And it said with those, if you plug it back in, then it'll fix the thing. I don't know if this has that same
Probably
ability,
not.
but
Yeah,
it's probably
that's
a
whole
lost
different
cause.
beast.
But
yeah, it can.
but yeah, I'll just push the stop button this time.
Yeah, that'll be your best bet. All right.
Okay.
All right. I think you're set. Awesome. You got video.
Let's see. So yes, this one is first, and that one is second. Okay.
I'll leave you to it.
Okay. Awesome. Thank you very much. Hello, my name is Brett Pike. I'm a grad student. Hi, I'm Brett Pike. I'm a okay. You, Darren. Hello, my name is Brett Pike. I'm a PhD student in the plant biology graduate group at the University of California in Davis. I work in Richard Nishamore's lab at the Genome Center, and my thesis project is seeking to edit the lettuce genome using a new technology called Cas9. So I would like to draw you a picture to tell you about it. So I am starting with a line of lettuce that glows in the dark, a red color, when excited by a particular wavelength of light. And this is because it contains an insert of a gene from a tropical coral. That gene is called DS-red. The species is it's a Discosoma species, and it's a red protein. So that has been inserted into the lettuce. Now what I'm trying to do is cause this red gene to glow green. And this by itself is not particularly useful, but if I can edit this gene, then I can use that same technology to edit any other gene in a way that would be useful for agriculture. I'll talk about that more in a minute. So the way that I do this is through a technology called protoplasting. And what that involves is taking a leaf of the lettuce plant, digesting it with two enzymes. One is called cellulase, and the other is called macerozyme. Plants do not have a skeleton that their tissues hang off of, but they do have cell walls, which cause the tissue to look something like a brick wall under a microscope with individual cells formed into square shapes, and each one containing a nucleus that contains the genetic material. These two enzymes digest away these cell walls, and what you're left with is perfectly round cells. Now these cells are very delicate because they're no longer constrained by this rigid cell wall. So they're sensitive to bursting, to small changes in osmotic pressure. But the advantage is that without the cell wall, I can now introduce foreign DNA to them. And because they have the DS-red gene, the cells continue to glow red under a microscope. So the DNA that I seek to put in these cells is a plasmid, circular piece of DNA. It's constructed synthetically, and is propagated in a bacteria, in an E. coli bacteria. So I can grow 500 mils of bacterial culture, and from that extract a large amount of this plasmid, which is a circular piece of DNA that has several genes in it. One of these genes is Cas9, which is an enzyme that cuts DNA, both strands of the DNA, double helix. And there's another gene for a guide RNA. The guide RNA gives specificity to the Cas9. It's a single-stranded piece of DNA that has 20 bases that form an exact match to a particular location in the genome. Now in this case, the gene that I'm targeting the DS-RED gene has an Asper gene at the 42nd position in the amino acid sequence. And so these Cas9 particles are programmed to cut very close to that 42nd position. And at the same time that I supply the Cas9, I also supply a single-stranded repair template that matches the DS-RED chromosome perfectly, except for one mismatch. And that one mismatch causes this Asper gene at the 42nd position to become a histidine. So this variant is called N42H. And this variant causes DS-RED to emit green light in addition to emitting red light. So I take this plasmid, which is much smaller than I've drawn here. I take many copies of it. And I aim to transfect that into the protoplasts, which have come from the leaf. I use an agent, a crowding agent here called PEG, polyethylene glycol, which occupies a lot of space in the solution. It squeezes the particles together, and it shocks the cells just enough that their cell membrane becomes somewhat permeabilized, and they're able to take up copies of the plasmid. Many of the cells die, about half of them die, but of the cells that survive, a good number of them should be transfected with this plasmid. So the way that I can determine if this process has worked or not is to use a technique called FACS, fluorescence assisted cell sorting. In that process, I take these protoplasts, I load them into the fax machine, and they go through one at a time in a sheath fluid, and it can do 1,000 per second at a very high rate. And so what this machine does is it has lasers in it, and it shoots each particle that comes through with a laser. And then it has a detector to see, is this lighting up in the red channel? And so because I'm starting with a plant that has DS-RED in its chromosomes, all of the cells will light up red. Now in addition, there's another laser, a green laser, that will indicate to a different detector if the cells have been converted in a way that they now release green fluorescence as well. This process is not very efficient. If I can get 1% efficiency, then that would be an excellent result and would allow me to continue with the process of regenerating plants. The advantage of this technique is that you can take a single cell and regenerate it into a whole plant. And if you're making any edits in that one cell, those edits will then be in the entire plant, including the floral organs, and will be transmitted to the next generation. So normally, when you make a genetic change, it's necessary to include a selectable marker, usually an antibiotic resistance marker, so that you can make seeds. If you have a plant that makes 10,000 seeds, if 30 of them are transformed, you can plant those seeds on a medium that contains your antibiotic, and the ones that sprout, you know, have your marker gene and in addition your gene of interest. Using this technique, if I can edit 1% of the cells, I can take those cells and I can plate them on Petri dishes, something called callus-inducing medium. So I put my individual protoplast on here, and I would wait for them to grow into microcalli. After the cell walls reform, they start to make little structures of undifferentiated tissue that can grow on a medium that contains the right balance of sugars and hormones. And once I have those microcalli, I can move them to a second medium called shoot-inducing medium that has a different mix of hormones in it. And from that, if it works, they will grow shoots, which I can then grow into whole plants and harvest seeds from that will be identical to the individual cells that I modified. This has the potential to be a transformative technique because it's not something that can be done without the use of Cas9. Because you need to know exactly where you're creating a double-stranded break in the chromosome in order to make a genetic change at that particular location. If I put this single-stranded oligodeoxynucleotide into these cells, I might get this conversion at a rate of 1 in a million. And this was something that was experimented with in corn and other crops in the 80s prior to the advent of modern genetic modification via agrobacterium that I'll talk about in another video. But at that rate, if you have a selectable marker, you can grow out 1 million seeds or 1 million microcalli and find the one that's resistant. But to avoid the use of a selectable marker, you need a much higher rate of transformation. So at the end of this process, what you have is a plant which is, in all other respects, exactly identical to the line that you started with, but now, instead of glowing red, it glows green due to this change in one base out of the 3 billion in the lettuce genome. So this is my proof of concept. And hopefully, if this works, I'll be able to do something more useful, such as putting in a gene from a wild lettuce that confers resistance to a disease that cultivated lettuce is susceptible to. That's something I'll talk about in future videos, but this is the basic project. Thank you very much for watching. My name is Brett Pike, and this is Public Domain.